Assalamualaikum, good morning and warm welcome to distinguished lecturers and officers, and also I believe there are students here. So my name is Fatin Ibrahim. I am the product manager from PLT Scientific Sdn Bhd, and I will be your host for today. Okay, first and foremost, I would like to express my gratitude by saying thank you for taking the time to join this webinar, where we will touch and talk about the total cell staining and advanced imaging for quantitative in-cell Western data. So with this webinar as well, we will touch on the Sapphire FL biomolecular imager, which is a laser scanning based system from Azure Biosystems. Okay, so we received a list of questions from you during the registration, and I hope the talk from Dr. Kai Leng today would answer them. But if you do have more questions or during the presentation, you have some inquiries, you're not sure about something, feel free to drop them in the chat box and we address them after the presentation, or you can unmute yourself towards the end of the presentation to ask your question. Just a little bit of introduction. So Azure Biosystems has been in the global market since 2013, and PLT Scientific Sdn Bhd has been their distributor in Malaysia since 2016. So Azure Biosystems have been developing and innovating imaging system that has superior flexibility and uncompromising performance. So we at PLT Scientific, together with Azure Biosystems, have been putting efforts to kind of introduce and bringing Azure's imaging system innovation to our local research community in Malaysia, so that in a way you would not stuck with the same brands or the same technology. And with Azure Biosystems, we're giving you more options to choose from. So with me today, I'm so delighted to have our speaker from Azure Biosystem itself, which is Dr. Kai Leng. So Dr. Kai Leng is the Regional Field Application Scientist for the Asia region and Oceania. Okay, so Dr. Kai Leng based in Malaysia, that is even great. So sometimes when we do some seminar or you wanted to do some demo, Dr. Kai Leng can be with us and support you in your experiments. So Dr. Kai Leng has been working with Azure Biosystems for more than 4 years. So I believe Dr. Kai Leng knows the up close and personal inside out of Azure imaging system. And she supports all the application and technical matters for customers in our region. So she has outstanding academic experience where she studied the molecular networks involved in brain maturation using the in vitro and in vivo models to study the Down syndrome disease. So Dr. Kai Leng also has an extensive research experience with all the molecular biology techniques you can name. So during this webinar, Dr. Kai Leng will be taking us through the principles of in-cell Western, the comparison of in-cell Western and the traditional Western blot, and also the tools that Azure Biosystems can offer you to perform the in-cell Western. So without further ado, I would like to welcome Dr. Kai Leng to give a talk.
Thank you everyone for joining today's webinar. So I am Kai Leng, the Regional Field Application Scientist of Azure Biosystems. So today, from the invitation from PLT Scientific, and today we will be talking about total cell staining and advanced imaging for quantitative in-cell Western data. So we will touch, this is very technical, but try to make it very briefly to guide you through the concept of in-cell Westerns, how to perform it, okay? Then we look into the necessities for you to get the data, of course, aside from your own cell lines and those criteria. Okay, and of course we're going to touch the imaging part as well, then guiding through our Sapphire FL, that is also for the high resolution laser scanning systems. Okay, right. So as Fatin introduced just now, Azure Biosystems is 2013, founded back in 2013. So it's a fairly new company. And then this year we have Sapphire FL, which is upgraded versions of our laser-based scanning system that we launched back in 2017. So it is a next-gen a fluorescent laser-based scanning system, very developed for Western for in vivo scanning, and also more others are high resolution scanning as well. And we have other equipments in our company portfolio. So you can see that we are quite focusing on a protein expression imaging. Therefore, we have a zoo imaging systems that is something like your gel documentation, but it covers chemiluminescence and fluorescence signals imaging. We have a qPCR system and also ChemiSolo, which is a personalized chemiluminescence Western blot imager. And on top of that, we have all the consumables for you to perform your Western blot for covering chemi and fluorescence Western blot. The thing that we don't have is just your primary antibody and the part that up to electrophoresis. So for imaging, we have three different solutions in order to see what you need to image. Okay, so we have a zoo imaging systems, we have ChemiSolo, and we have Sapphire FL biomolecular imager. So these are the applications that these three that covered by these three uh, equipments. Okay, so let us see one by one. Okay, so the first will be Sapphire FL. Okay, so Sapphire FL covers the high resolutions part of the fluorescence signals imaging, including anything that fit within 4 cm, like your tiny mouse or skinny rat, and phosphor imaging as well. So this will be the part that we gonna use it to scan your in-cell westerns, either be a 96 well plate or 384 well plate. Okay, so that is why we're using Sapphire FL. It can reach the higher resolutions imaging. Then we have zoo imaging systems, especially the 600 models, which is a higher specs model, where it covers the fluorescence signals that you can get in basically blot on gel or other how to say resolutions that is reaching for 40 micron. Okay, but most importantly, all of them can do your basic gel documentations like nucleic acid gels and protein gels and colorimetric signals detections. And we have another models and onwards 300, 400. Okay, onwards they can do chemiluminescence, then optionally fluorescence signals detections. And of course, ChemiSolo, which we dedicated to have only chemi signal imaging. All right. So here we look at the equipment parts about how to image your in-cell Western. Let us look at what it is instead. Okay. In-cell Western, we have been listening to this term for many, many years. I remember back in the days. Okay, so they've been talking about like Western in the plate. It can be said as a cell-based ELISA. So what it is in truth, it is a very powerful immunofluorescence assay. So we know that it uses your primary antibody and your secondary antibody for detection methods, and it is a very high throughput solutions to quantify your target proteins in cultured cells. So there are a few keywords in cell westerns here. So firstly, of course, is immunoassay. It requires primary antibody. It's high throughput. So here, high throughput means you have every well representing one data point. Okay, then by utilizing the primary antibody allows you to quantify the expressions of your target protein. Okay, by it be let's say pigmentations, by it be a cancer marker, by it be any wound healing marker, any markers that you deem it able to for you to QC or able to how to say to analyze and give a solid answer for a certain questions. Okay, and this also need to be done in cultured cells. So the cells need to be adhered in the plate. Okay, and then of course we have fixing. The steps are basically like your ICC, immunocytochemistry. Okay, but it doesn't the way it interpret the data is different than ICC. So that is the in-cell westerns. So the process goes like this. You have cells where you plated or you seeded okay, seed them overnight, then you fix them, either you grow them, depend on the conditions or your experimental design, then permeabilize in the wells. Then if you're detecting the proteins inside the cells, that will be in-cell Westons. If you're detecting the protein on the membrane of the cells, that or the extracellular matrix of some sort, that will be on-cell Westerns. Okay, but in the term, it's something like in-cell Western, on-cell Western. Irregardless of that, they are always in the format of microplate, like 96 well plate or 384 well plate. So it's an immunoassay. So you do need your target specific primary antibody plus a labeled secondary antibody. Okay, so how we come up or how the community come up with this idea about in-cell Western is it combines the specificity of a Western blot with the high throughput of ELISA. So we are combining the perks of ELISA and the Western blot. So we want the best of two. So we know that most of the lab knows how to do Western blot right now because it has been the very generic technique for us to do protein expression studies or protein discovery. And ELISA, we use it more about, we want to know about the minute quantity and then the changes across the time. And also we want to, plot a certain graph of some sort. So they have been, these two techniques has been in the lab for many, many years. Okay, so let us look at the perks or what they can do individually. Okay, so we will be talking about centralized around this graph. Okay, right. ELISA, we know about it, is also an immunoassay. Okay, so you analyze the cell lysate or any other lysate or mixed product from your samples. Okay, urine, blood, or something like that. So that will be your samples. It is high throughput because it's mostly the antibody, either be a sandwich or non-sandwich manner, because direct or indirect, there will be the antibody will be prop on the plate. So we just need to see it, or put your sample in, then you can get the results. So it is high throughput. It comes in a manner of 96 or 384 well plate. Okay, so it also comes with um some depending on the assay or kit, you might get able to get like a multiplex kind of data, like two or more or three kind of data, protein data in the ELISA, depending on the kit that you purchase. Okay, so that is what we normally know about ELISA. It's quick, it's really quick. Okay, it doesn't involve any other kind of how to say multi steps consumables. No, it's a very straightforward techniques. And Western blot, okay, it is a multi-step techniques. So we have gels, okay, electrophoresis. Then we go transfer the protein to your membrane. Then we go through blocking, primary, secondary, and some sort. Okay, and then we visualize the band. So the data comes in the band. Okay, so we assume that the band will be will give you the band intensity. The band intensity will reflect the protein amount that you want to see in the sample. Okay, right. So it's a multi-steps techniques. Okay, and you also need to have your sample purified, centrifuge, like so on. So, but it is very low throughput. As we know, it's very low throughput. If you have 4 or 5 protein targets, and let's say you have 6, n equal to 6 for every group, you definitely have more than 3 groups. You need to have one non like negative control, one positive control, and one of your treatment group. Okay, then this is how you're gonna arrange your blot. You're gonna arrange it in a more proper manner, like representative what is a sample from every group, and you have to do technical replicates. So it's gonna spend a lot of time. So by the end of the day, you do a lot of Western blot. Okay, so that is very low throughput compared to the ELISA. In the Westerns, so what it does is like this. Even though the concept or the steps are very, very similar to ICC, but in immunocytochemistry, but the concept of analyzing the data is different. Okay, so let us look at here. So we call it Western. So we compare directly to Western then. So this is a format of in-cell Western. You fix the cells, okay, up to 96 types, cell type or 96 wells, up to you. In 96 well plate, we say 96 well plate, okay? But in the terms, you can do any microplate, okay, as long as you fit the dimensions. And you permeabilize the cells, probably primary, secondary, okay, for one or more proteins. So it usually utilizes the fluorescent signals for you to do multiplexes because as we know for Western blot, you do need to do normalizations. Okay, so it's the same concept. In Western, you need normalization. Same goes with your Western blot. So for Western blot, you load the samples into a gel. Okay, then you transfer it to a membrane, primary, secondary. It's the same thing, but you see the difference in how to say the amount of data point that you can obtain from a single run. Okay, so here let's say I have like 1, 2, 3, 4, 5, 6, maybe seven groups. So in a single plot, I can mostly put 1 or 2 samples from every group. And I do need to do technical replicates, mean I need to repeat the Western blot techniques twice. Okay. And for in-cell Westerns, let's say the single treatment or single sample, you can load it like 6 wells, depending on how much you want to fit in the single plate. But ideally, everything should fit together, including, I mean, everything from your experimental design should be included in a single assay. That's the control you want to go for. Now you repeat it for your technical replicates times 2. Okay, so what's the difference here? The difference here is here, you may get 1 or 2 data point, okay, per technical replicate. Here, you can get more of that, okay, maybe 3, maybe 6. So this is what we say high throughput, because by having more data points, your variation of error is reduced, okay? So that is the main difference of that. Next thing is when you need to do your in-cell Western, okay? Right. Okay. Hi, sorry, can you mute your mic, please? Right, so it's slightly loud. Thank you. Okay, right. So when you need to use the in-cell Western and when you need to do your Western blot. So firstly, when you use the Western blot is you need to when you need to do your high throughput analysis of characterized proteins. Okay. Hang on, let me see what I can do with the hello? I can't really mute. Okay, thank you. Right, so all right, come back here. Come back to the slide. So when to use in-cell Western is when we need to do high throughput analysis of characterized proteins. Okay, right. So this is where you know what you're looking for. What is the protein target you're looking for? Okay, for like what I say, when you use it is for drug screening, molecule like bioactive screening or active compound screening or pharmacological screen for any kind of like I say active compound and so on and so forth or medicine and drug. So you know what kind of target you're looking for. Like say anti-pigmentation, like whitening products, or let's say like anti-cancer properties or anti-inflammation properties. So you know the markers are looking for will be an inflammatory markers, cancer markers, or pigmentation markers. So these are the markers you already know. They allow you to quantify, like let's say, allow you to access whether the let's say the bioactive compound have these properties and until what extent. So that's why then you can do it in a large scale. Okay, so mostly you in pharmacologic like biopharma company, but nowadays it's very commonly used in research as well. So Western blot, the very generic technique that we know, it has been very commonly used to, we look at it, we have the band, we look at the molecular weight, know that that is the protein we want to see, and then we can identify a novel protein or discover a new proteins, let's say, we can also look at like say post-translational modifications, that kind of studies. Okay, so that is a very, it covers of all the generic things about fundamental research that is Western blot. That's why it's a very common use in academic research and discovery. So here's a table to compare between incel Westerns and traditional Westerns. Okay, so both of them can allow you to do protein profiling, expression profiling, means you can access the protein expressions, and you can do multiplex analysis. Okay, you can do more than one target in the same run. Okay, so in traditional Western, that will be something like fluorescent Western blot then. And it also gives you quantitative data. Okay, this will be also built on the context of fluorescent signals. Okay, therefore you can get a quantitative data. Okay, so both of them share these properties, but traditional Westerns allow you to discover new or novel proteins or discover new kind of modifications and molecular weight analysis. And something that traditional Western could not give is high throughput data generation and also in situ analysis, means the data that you get is without disrupting the cellular environment, because if you want to perform in situ analysis, it's very difficult. Either you need to culture the cells, try to observe the expression changes when they are alive, okay, live cells imaging, that kind of situations. Or else, most of the time, you would need to harvest the cells, trypsinize it, lyse it. So therefore, you can't really see the cellular environment. It always has been lysed, trypsin lysed and then ready in lysate forms for you to do your traditional westerns. Okay, so that is another perks for incel westerns. Okay, so now we know that both of them, okay, incel, you decided to go for incel Western, okay, right? So you want to look at your cell signaling pathways or biology studies or screening, drug screening and biomarkers. So normalizations, you need to choose a normalization controls, much like your normal Western blotting, okay, right? Ideally, okay, here's the concept in incel westerns, the numbers that we can control is always the numbers of cells. Okay, so we control the numbers of cells we seeded in every single well. You can do in increasing amount of cells, okay, for you to plot a standard curve, that's fine. But that is also a control manner, is a controlled. So we control the protein concentration that we loaded in the gel, SDS page gel in the traditional Western blot, and in incel Western, we control the numbers of cells. So in reality, even though we try to, standardize how much cells that we seeded in, but do remember that we have different treatments, we have different, even different culture conditions, okay, even different seasons. So it may not apply in Malaysia, but it did apply to certain other countries. This would change the data that we obtain by the end of the day. Okay, all these ambient environment would change. As much as we want to control it, it will change. Okay, so sometimes how we gonna, how to say, interpret the data, as you can see the graph like this. Okay, so the protein volume, okay, so we here will say protein volumes up and down, and then but it doesn't really reflect the expression changes that we see, okay, based on the cells number. For example, here the cells numbers are higher, but the expression level is low. So how do we explain this? Are we gonna say, oh, it's because they die a lot or some sort? So there are different criteria for you to actually justify, okay, and say that, or is indeed having, let's say, an anti-cancer properties, something like that. Okay, so we need a normalization control. Okay, so much like our Western blot, incel Western also having the same thing. Okay, so after normalizations, okay, we know that uh, so here the normalization will be accounted for the difference of number of cells between the wells. Okay, so we are using the number of cells in the wells as your normalization controls, because that is what we know. Okay, right. So you can see that after normalizations, okay, that we will know that the volume will be more accurate because it's based on the sales number. Okay, right. Let us look at the difference between the traditional westerns and in-cell westerns. Okay, right. The normal western blot, we always have normalization that is within every single lane. Okay, so every single lane, because they are from the same samples, so either be a target protein or either be a total protein. So it will be like lane normalization, total protein normalization, or something what we call a housekeeping normalization. So it will be something in that sample that we use to normalize, something that we deem that it will not change or deem as much control as possible. And for in-cell westerns, it usually normalize against the total cell signal within every single well. So because it is a cells, so we don't look at individual cells expression, but we look at collectively the expression in a single well. So since we're looking at the signal intensity within a single well, so something that to represent the entire well will be good enough for your normalization control. So just like our traditional westerns, we have two types of normalization in cell westerns. So the first thing will be your reference protein, something like a housekeeping, we call it housekeeping, like a GAPDH or actin, and so on and so forth. So the pro is it is very biological relevance because you can choose a similar protein that you know of. Normally, that is like FDH or actin or some sort based on your research, and then you can choose a range specificity. So either be high express, moderately expressed, or lowly expressed. So this is something that you can choose where you know that they are behaving like that in the treatment. Okay, but the bad thing, like the reference protein or housekeeping protein as a normalization control, either be in cell westerns or in traditional westerns, is we need to assume that these housekeeping proteins are expressing constantly throughout the entire samples. Okay, but we know that it is not true, and it doesn't really follow that trend because, as I mentioned, we have different treatment, we have different sometimes human errors, sometimes where you see the cells, how you wash the plate, okay, where you put at the side, put at the middle. There are many, many ways to actually change the expressions. Okay. So that's why there is another type of normalization, which we usually deem it as more controlled, is a cell stain. So cell stains is they don't care about the expression of all the changes of the expression that you get in all these cells. They don't care about it. So we care what we need is how much cells in that well. And then all this expression we just normalize against the cell's number. So it's usually staining a DNA or total cell component like your cytoplasm. Okay, so there are many types of staining that you do. Okay, so we want to remove for cell staining. It enables or it allows the user to remove the potential for all the normalization control to influence by your conditions of treatments and all the other things that you cannot foresee, like say treatments and human errors or different incubators, all that kind of thing. Because right now, by the end of the day, you are looking at the total sales. So the sales is high, the sale is low, the sales numbers are high or low. That's it. That is it. You normalize against that. Okay. So the expression changes of the proteins, it doesn't matter right now. Okay. So right now the numbers of cells are considered based on the condition, yet high or low. That's it. Okay. So the dynamic range is much higher because you have the number of cells you know that you seeded in. Okay. Then you know that how much cells has been, let's say, died during the treatment, allows you to get a mortal like ID50 or some sort, and you can tweak around the cell numbers, allow you to reach the point that gives you a very reliable result by repeat. Okay, so you do need some what do you say, optimization before that. Okay, right. Both reference protein and cell stainings, these two are working great, but they do have their own pros and cons. Okay, right. So this limitation for cell staining will be the cell numbers because you need to tweak around the cells numbers in order to not losing too much and then able to analyze it properly. And the cons for this reference protein will be the assumption of constant expressions. Right. So in cell Western normalization, there are I'll say 4 or 5 types of major in cell Western normalization we see on the market right now. So from Azuba systems, we have offer Azucyto. So it is an in DNA stain, so it stains cells, okay, the DNA. So it image in your Cy3 channels, and it has the per kit it can allows you to stain for 596 wells. That is the kit including 2 secondary antibodies plus all the associated assay reagents. Okay. And then there are others in cell Western normalization that staining nucleus and cytoplasms, or like azocytol staining the DNA, or housekeeping like a housekeeping protein normalization. So this is housekeeping normalization. And you will see that why they are very quantitative is they are always in the fluorescent signals detections, like in order like Cy3 or infrared 700 or Cy5 on all these channels. These fluorescent signals are quantitative signals. So we need it in cell westerns. Okay. Right. I see hands raised up. Habibu. Please. We have a Q&A session after this. So you can raise your question in the Q&A sessions, okay? After the presentation. Thank you. Right. So this, if you wanting to do your in-cell westerns, so you can screenshot this right now. Okay. Then you quite to see the price point. Oh, sorry, I do and also the channels you have in your imaging systems, and then what kind of normalization you want to go for. You can try and error on that. Okay. So what we have offered is a ZooCyto is an all-in-one package for your in-cell westerns. Right. So here's the steps for in-cell western. All right. So like all our research analytical techniques, every single steps to be QC, okay? They should be QC properly. And we have your primary antibodies, okay? You have your secondary antibodies, total cell stains, okay? And we have wash, fixation solution, permeabilization solution, and blocking buffer. So this you can see the steps are basically almost identical to your ICC immunocytochemistry techniques. All right. So, okay. So here we have like a fixing solution. All right. They fix, they permeabilize solutions. Okay. In the culture dish, then you do your blocking. Okay. Prevent unspecific bindings. Then incubate your primary antibody. Okay. Then you wash, then incubate with your secondary antibody and all your total cells stain together, right? And for in-cell Western, normally the secondary antibodies, we usually, for your target protein, we usually do it in near infrared fluorophores, like your uh, infrared 700 or 800 channels, because these channels or near infrared channels, they are less subjected to interference from the autofluorescent signals or the noises from the plastics. So that's why our kit actually only perform in 700, 800 for your target proteins. Okay. Then you have a total cell stains, either be a nuclear stain, cytoplasmic stain, that will be used to normalize the signal from your in-cell Western to the number of cells in the wells. Right. So this is our Azul Cyto in-cell Western kit. As you can see here, it contains all the critical reagents to perform an in-cell Westerns with detection of 2 targets in the near infrared channels, 700, 800, plus a total cells normalizations. Okay. So it is the kit contains all the reagents necessary for staining a whole cells for total cell normalizations, okay, plus simultaneous detection of 2 biomarkers for your 5 times 96 in-cell Western assays. Okay, so this the whole set is already QC for in-cell Westerns. And do remember that we cannot most of the kit doesn't include your primary antibody because you need to validate the specificity of a primary body in your experiment. Okay, right. So here, let us look at the Azul Cyto in-cell Western kit. It has a permeabilization solutions. Okay, it has a blocking solution, total cell stains, and secondary antibodies. Okay, these are basically the reagents that you need after you seed the cells. Okay, overnight they are nicely done. Okay, and then there you go. And then the fixing, so this is without a fixation, so you need to fix it either with PFA or some sort, depending on the, how to say, different kind of fixations based on your experiment designs. Right. So here's look at the steps. Okay, first thing. So if you look at it, it looks very much like your ICC. So you see the cells in 96 per plate, incubate overnight. Okay, let it sit properly. Then you fix the cells. Okay. Then the kit allow you to do permeabilizations, blocking, incubate with primary antibody with the total cell stain, wash, secondary, which is in our near infrared channels, 700, 800. Then wash away. Then you can scan or image the plate in 5, this is side 3, and near infrared 700, 800 channels. Okay, so this is the whole protocol for in-cell Western kit. All right, but it's applied to all your in-cell Western as well. Okay, so the thing is, in if you develop your own in-cell Westerns, you can do so. Okay, but do remember that you have to QC your primary antibody first, and you do need to generate, if you want to perform by yourself. Okay, you need to do a standard curve to show the linear signal linearity. Okay, according to the cell number versus the certain number, a signal versus the cell number. Right. So azucytol total cell stain. So here's the example of the total cell stain and plus the signal that we detected in 700, 800 channels in a cell that we seeded in dilution manners, right? So what it can allow you to have is the signal linearity. First thing, the total cell stain is linear, is linear. What it means by linear is the fluorescence signals, okay, is linear versus the number of cells. So you can see the range of these total cell stain is by 225,000 numbers of cells in the single wells. This is the R square, so showing that the signal linearity is good until 25,000 of cells. Okay, so for this applies to your total cell stains or any other normalization control you want to use in your in-cell Westerns. Okay, you need to show the signal linearity versus the number of cells. Okay, so you know that, yeah, the how much the signals representing how much the cells you have in the wells. Right. So second thing is it casts the sensitivity. So you not really worry if you have a very low amount of cells in the wells, let's say a few hundred cells, because the signal linearity covers it. So we image the entire thing, and how you analyze the signal will be every wells is one data points. So we are not looking at the cellular levels. We are looking at well levels, means one wells is one data points. So your imaging equipment, as long as it can image it reliably, a very good signal linearity, clean background, okay, then you can plot a very nice curve or like representing the protein amount that is good enough for your in-cell Westerns. So in reality, normal research lab is capable of doing in-cell Westerns. You do not need high, how to say, very very good, like very expensive equipment to do so. No, because by the end of the day, as long as the signal linearity, your analysis, all the data, the signals is fall within the signal linearity, that is true. And of course, your normalization must be good. Your control must be negative and positives are good as well. That means your assay is valid. Okay, right. Enhanced accuracy. So for this enhanced accuracy is because we utilize the total cell stains and also the fluorescence signals from 700, 800. So it is a very good tools for anyone who want to screen at a large scales but they want a clean data, and they want a very crisp and want a very good signal linearity. Okay, right. So here's about Asus Saito, total cell states, and also the entire in-cell Western skid. Next, let us look at the equipment part. Okay, so you do need to image the in-cell Western. All right. So as I mentioned, okay, Sapphire FL, it can reach up to 5 micro resolutions. It's a high resolution fluorescent signals detections. Okay, so if you're performing in-cell Western, first thing you need to check is, do you have equipment to scan or to capture the signals from the plate? Okay, they need to have a resolutions that is good enough to separate the wells to wells, especially if you're doing 384 wells. Right. Sapphire FL, so FL stands for fluorescence and flexibilities. So we know that it can scan your fluorescence signals. Okay, right. So it has these adjustable Z plane up to 5 micro resolutions, 5 anesthesia port for live animal or live organism imagings, and also customizable and user changeable optical modules. Right. So let us look at how it detects the fluorescence signals. Sapphire FL is our how to say our highest end uh fluorescent imaging signal fluorescent imaging equipment in Azuba systems. All right, so this is how it looks like. Why is the best in our equipment is because it uses PMT and APD, but not CCD and CMOS for your fluorescent signal scanning, right? So these two are the best for fluorescent signals detections. All right. So it gives you super clean background and gives you the resolutions you want to go for. Right. CCD is very good for your fluorescence blot, fluorescent gels, or fluorescent plant or biomarkers, or very preliminary screening on your slides or well plates. That is good enough. But if you want to do something like 384 well plate, or you want to have a very clean 96 well plate result, then Sapphire Alpha will be the choice that you go for. So how it does is it is utilizing a laser scanning. So it has lasers, okay, excitation light sources, and you have a PMT and APD detected. They give you point to point or pixel to pixel scanning. So excitation scanning, point to point, pixel to pixels. Okay, so it is very sensitive. All right, and then it's very we have able to scan and separate 384 well plate very nicely and reliably. Okay, so Sapphire FL, we know that it's focusing on fluorescent signals, and the next thing is the flexibility. So why the flexibility comes in is to accustom to different kind of dyes that we usually see in the laboratories. So as we know, we have uh fluorescence channels. We have 5 major RGB in the visible fluorescence and the near infrared 700, 800 in the near infrared channel. So we have total 5, red, green, blue, and also the near infrared 700, 800. The red, green, blue can be your Psi 2, Psi 3, and Psi 5. And the IR 700, 800 will be your like Psi 5.5 and Psi 0.5 and so on. Okay, right. So that is the generic fluorescence channels that we see, and we do know that in confocals and in fluorescent microscopes and other kind of higher end equipment, you normally have something like a yellow fluorescence protein or PI or M cherries and DAPI that you use to stain your DNA or cells
Update.
for your ICC. Okay, right.
Yeah.
So this is for
Yes.
us Sapphire FL.
My name is Whitbina.
Oh, sorry, Nicole, I can hear your mic. Okay, thank you. Right. So this is Sapphire FL. We have 1, 2, 3 slots for you to slot in your optical modules. So it comes the optical modules have its own. This is optical modules. You have lasers and all the detectors, and plus this is tiny thing. This is your filters. Right. So this is an uncoupled design, right? So the lasers and emission filters, they are uncoupled. They can swap around okay, allows you to perform or image your yellow fluorescent proteins or PI. So you are not necessary just to only able to use the system for your standard RGB and near infrared channels. No, it can be used for any signal detection as long as they are we able to how to say to produce it. That's it, right? So these uncoupled designs, first thing, allow us to have DAPI, right? So DAPI, as we know, is a UV range excitations, okay? And this is what the laser-based scanning system right now on the market. We are the Azure Sapphire Alpha is the only one able to image DAPI from your samples, right? 375, okay? And emission filter at that 452 range. Okay. So here is the data that we have in-house for to show that the DAPI module is functional, right? So we have in-cell Westons of a HeLa cells in serially diluted in 1 to 2 dilutions down the columns, right? So the bottom row is your negative controls, right? The cells are staying with DAPI. Though the DAPI is very cheap, you normally use it in your ICC, all right? Okay, so this is the same plate where we have stayed with DAPI and the HeLa cells, and we scan with the standard 488, the optical module. You can see we can't get any signals here, all right? So the signals are not detectable in the traditional 488 lasers. But with our DAPI optical modules, you're able to get the DAPI signals in a very good signal linearity manners because we have it in 1 to 2 dilutions. Okay, so that means that our DAPI optical module allows you to detect that DAPI in your samples right now. Okay, so as I mentioned before, this is uncoupling excitation designs, excitation from emission designs, so you can mix and match. So this mix and match, one of the perks is allows us to use or detect mega Stokes dye. You will start to see this dye on the market very soon, or they're basically on the market right now. So what are the differences? The standard FIT-C, Psi2, Psi5, Psi3, you can see the Stokes shift, okay, they are normally within like 50 nanometer wavelength, okay? So ideally, we want the excitations and emission spectra to be separately enough so they don't bleed over to the detection channel here. From the excitation, they don't bleed over to the emission channels. So here's a bleeding here. There's a bleeding. Okay. So this is a mega Stokes dice, the spectral separations. You can see, so there are a lot of R&D and discovery looking for dyes. They have a very big Stokes shift. Okay. Stokes shift is the distance between the excitation maximum, excitation, sorry, and emission at maxima here. This is Stokes shift. So normally it's between 10 to 40, but mega Stokes dies, they have up to 80, more than 80 nanometer wavelength, where it can minimize the crosstalk between the excitation and the detection. Okay, less bleeding, so give you a very, very good clean background plus signal to noise ratio. So these are very good dice that you can use, okay, if you want to use it, utilize this in your research. Okay, it's basically from chemical or physical properties eliminates or minimizing the crosstalk within the fluorescence dye itself. So this is basically the mega Stokes dyes. So we use it on our fluorescent Western blot where we have a HeLa cell lysate probe with the STAT3 primary antibody. And you can see because it's an uncoupled designs, we can pair the laser with different emission filters that allows you to capture the signals from these mega Stokes dyes, DY481 XL mega Stokes dyes. Right. So Sapphire Alpha, we look at it, we know that it scans your fluorescent signals, is very flexible. Then we look at how flexible in terms of the dyes that you can scan. Next, how it applies on the sample and down to our in-cell Western will be, it has adjustable Z plane up to 6 mm for to fit all the samples that you can think of. Okay, and also have resolution up to 5 micron, which is which is normally we use it for like 384 well plate and glass slide or tissue slide imaging. Okay, right. So we have different focus, so you can scan your membrane, gel, and even your plate. Okay, now so we have like inbuilt software inbuilt function for you to create a GIF file for looking at the expression and different plane, and also a merge images from different Z plane. So this is like a Z stacking, okay? Like you image it in the confocal. So it comes since we can capture signals in different depth, so it supports a variety of your studies. Okay, cover from the blood, okay, your plate, tissue slide, whole organ, and live animal. We also the Sapphire FL also have two types of sample holder available, the plate holder and a slide holder. Here is the plate holder where you can select from the protocols, and it will automatically plot the scan regions on the software. So what you need to do is just select the resolutions and also the focus. No, the focus is already preset, and you select the lasers. Okay. So here's an in-cell Western data that we have, where we have the plate focus setting at 3 mm. All right. So here's we have the HeLa cells that we have in seeded in 1 to 2 serial dilutions, right? So we have in the blue pseudo color, it will be azul cyto, with staining the DNA, representing the normalization control. We have 700 channel in green, okay, pseudo color green, that is staining the human, I think it's human, RMPK, and GAPDH in infrared 800 channels. So this is three-plex in-cell Westerns, okay, and scan in Sapphire FL. So the Azure Cyto in-cell Westerns are basically optimized and validated to work together in Sapphire FL. It's basically optimized around Sapphire FL to give you a very clean background. Okay, you can see we have these application notes here. Okay, we have different. You can see here if there's no background, the background is very clean because of the laser excitation light sources and the PMT and APD detectors. So they give you very clean and very huge dynamic range. So that is very ideally for your in-cell Western scanning. Right. So moving on, a lot other than your in-cell Western scanning, Sapphire Alpha also, because it can reach up to 5 micron resolutions. So we can use it to scan your tissue slide. This is a mouse lung tissue. What it gives the data is a whole FOV or field of view of your tissue section slide. So of course, normally we do stitching in confocal or fluorescent microscope, and for Sapphire FL, it scan everything in a single shot, and so it gives you a single field of view. And with the software, allows you to burn in the scale bar where you can use it for your manuscript publications. Okay, manuscript writing or publications. All right. So we also can scan more slides here. This is more to show you 5 micron resolutions. Four-plex fluorescence Western blot. Okay, this is fluorescent Western blot. It's a quantitative fluorescence Western blot. Very clean background. Fluorescent gel imagings. Okay, so you can use it for, let's say, your bacteria that is has like GFP tag or some sort or certain expressions that you can just load into the gel and scan on the Sapphire FL. Comesi Blue gel imaging also capable to be scanned on Sapphire FL using the near infrared 700 channels. I say 700 because it's 685. It's nearing 700, right? So this is Comesi gel imaging. Cell-based assay, for example, we have 488 and the PI, okay, for you to quantify the viability of the cells within the wells, and of course, more about the yellow fluorescence protein. This is what we tested to check which kit is the best for our transfections. So here you can see this is 24 well plate. All right. In a single field of view, you know that which kit gives you the best transfection efficiency. Then you can pick the one. For example, this is the best one, right? And of course, the other checking of the liquid culture of the cells expressing red fluorescent proteins, right? And tissue array to look at multiple tumor matched tumor and normal tissue microarray scanned at 10 micron resolutions. These tissue are basically 1.5 mm in diameter. It's a very, very small. That's why we need to scan in 10 micron resolutions. So basically it has hot trace blue, okay, scanning the DNA and nuclei. This is basically like a housekeeping or normalization control or just a passive like reference. And you have 488 for the high affinity filamentous actin. Sorry, for staining the F actin to show the tumor marker. Right. Then the Sapphire FL also has 5 anesthesia port, gaseous anesthesia port for to image your live animal. So here we display 6 here. So because they need three, okay, to have a control for their mouse. Okay, so these are mice. So there are 4 cm clearance here. So anything fit within 4 cm, you can scan it. So what they have is over here, there will be a tumor growth on the side on the glass scan bed. So the scan bed is 25 times 25 cm. So you can place quite a lot of things on there. Right. So scanning the live animals, so what it can do, what kind of studies that you usually utilize this function will be preclinical drug screening, tracking, okay, cell migration study or tumor growth. Okay. So this is a Sapphire FL serves as intermediate steps before they go use like they before they scan these in a very high expensive and bulky instruments so they can check and optimize their protocols before they use the high-end equipments, right? So this is an image just to show you that shave off the fur, so remove the autofluorescence signals. Sapphire FL will give you a very, very clean signals. So this is what we try to show here. Right. Next, let us look at the tumor imaging. So these are the tumor cells, okay, 41 cells with red fluorescence protein injected in the subcutaneous area. And then we image after 11 days, and this image is captured at 100 micron resolutions, okay? Right. So we have these 4 days post injections, 11 days post injections, and then 14 days post injection. This is ex vivo. Harvest it to tally the size of tumors that we get see in the body. Right. And this is a GIF file, the GIF image file that we produce, okay, from the different Z plane scanning here, from 0 up to 3 mm, each scan at 100 micron resolutions. Okay. Right, so you can see the expression of the red fluorescence protein, and these are having the tumor but without the red fluorescence protein. You can see the signals are very clean. Okay, right. So so all these fluorescence signals detections also can be imaged in extended dynamic range. What it means is now instead of saturated signal where you cannot analyze the data anymore, if with extended dynamic range, you can fit high expression samples like overexpressed samples and knockout or knockdown samples and your wild type, everything at the same on the same time, scan at the same time. And with extended dynamic range, it take slightly longer time, but it will give you a image that you can analyze. Okay, so the idea is if your signal is saturated, you cannot use it for analysis, right? So that's why we have this function for your fluorescent signals. Here's an example. If under normal conditions where you have highly expressed proteins, they are usually saturated. But with the extended dynamic range, you extend the dynamic range, now it can fit all the high signals, high intensity signals, and low intensity signals at the same time. Therefore, you can analyze this data in the software. Right. So FireFL also have phosphor imaging module. So to image your radioisotope labeled samples like your hydrogen 3, carbon 14, phosphorus 32 or sulfur 35. So these are normally used in, let's say, metabolite studies like protein tagging or protein or lipid, or glycoprotein or polysac or nucleic acid. So this is Sapphire FL capable of doing as well. And of course, it has a very good signal linearity. So for those who wanted to do in-cell Western's, but they don't have a gel dot, they wanted to have chemi signal imaging. We also have a chemiluminescence module as a site, a small it's a small equipment, site equipment that is used together with the Sapphire FL. So the chemiluminescence module allows you to image your chemiluminescence signals, and of course allows you to do densitometry analysis where you quantify the spots or bands based on their optical density on your colorimetric stain protein gel, like your Comanci or silver stains gels, and X-ray films as well. Okay, like your northern blot, southern blot, or chemiluminescent westerns. So this is densitometry analysis using 21-step tablets, okay, using optical density to plot a center curve. There you then quantify the one on your unknown gel or unknown samples. Right. So here's the Sapphire FL, where it has flexibility in dyes and fluorescence signals. Okay, sample and applications. We have a lot of application notes. Okay, depending on what you want to see, we have it in tumor cells. You can see the application notes here. Okay, tissue slide in 5 micron resolutions, whole slide imaging, live animal under anesthesia imagings, cell viability assays, okay, visualization of PI, okay, and also yellow fluorescence protein expressing cells, okay, uh, mix and the customized mix and match laser emission filter allows you to image a mega stock dyes, okay, and BioFC assay, biomolecular fluorescence complementation assay, for your in vitro protein ligand interaction studies that perform in 24 well plates. Also, we have application notes on that. And if you want to check out our publication, we have a lot of publications right now. It's been booming since this year and last year's. And we have publication booklet as well. These are application notes. And in summary, so Sapphire FL, because of the adjustable Z-plane and also the focus and high resolution scanning, we able to use it very reliably for in-cell Western scanning. So it has been very well received. Sapphire FL, these are our user for Sapphire FL, even though we just launched end of last year. So they have already purchased and having the system in their laboratories. And here's our imaging solution for every labs and applications. Thank you for your attention. We shall open the floor for Q&A sessions. Okay. Right. Let me. Oh, okay. Sorry, I forgot to mention. Right. So, for Sapphire FL, okay. So for every single 96 well plate or 384, the dimension they scan, it only takes at 100 micro resolutions. It only takes like less than 7 minutes to scan. That is why we 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 kind of focus on in-cell Western because it is very high throughput, it's very quick, and then everything from the scanning, from the techniques itself, the staining up until the imaging. Okay, thank you for attention. I'll pass the floor back to Fatin.
Yes, thank you so much, Dr. Kailing. That was a very informative, I would say, presentation where you walk through us about the in-cell or the comparison with different techniques like the Western and also the immunofluorescence. So I just would like to know if there's any question from the participants attend today. Do you have any question?
Hi Fatin.
Yes.
I heard that you say some of them submitted question when they register, right?
Yeah, okay. But the participants were not here, but one of the question was like, what is it? Like, what is in-cell Western? which I believe you already explained that in
Okay.
your presentation. Another question is from, let me see, is from Shalini Tatu. I don't think she's here, but she was asking what is the mechanism of detection of biomolecular imaging?
Mechanism of detection. How it works, is it?
Sorry?
A mechanism of detections?
Yeah, so I believe what she's trying to ask is the detection method that we use. So I believe it's the laser, PMT, and APD.
Yeah, it's a point-to-point scanning system, laser scanning system.
Yeah. And another question is from an officer from FRIM. So
Mm-hmm.
she was asking, I would like to know this instrument's ability to capture cell culture image, that is anti-tube formation assay or anti-migration assay, Hirsch assay.
Mm-hmm.
And she wanted to know if you can do, Sapphire FL can do real-time based or recorded video.
Real-time based recorded video. Unfortunately, no, because it's a one-time scanning. So it's one-time scanning. You can just keep the plate. But the thing is, because you want to have it alive, so
Mm.
you need to have incubator. So for Sapphire FL, it doesn't have incubator for that. So it's not served as a live cells imaging. It's more like it does support live small animal imaging,
Yeah.
but not live cells imaging. That's why the in-cell Western comes into picture because we can fix and then permeabilize and then we can scan it within 7 minutes for a single plate and then in a quick manner. Yeah.
Yeah. Okay. Thank you so much, Kailing.
No worries.
Do we have any more questions?
Oh, I think we have a question in the chat box.
Yeah. Hi, Dr. Kailing. I am interested with the application of Sapphire in determining the in vitro protein ligand interaction. My current study is development of antiviral drug for marine fish against beta nodovirus. Can I
Mhm.
use Sapphire to test the interaction of my antiviral and the viral proteins? Thank you so
All
much,
right.
Ms. Hanis. Yeah.
Hi, Miss Hanis. Thank you so much. So that I think you are interested in the Bifi FC essay that I showed just now, right? So if you're interested, I think I will let our can you leave us your contact so we can send you the application notes? So yes, you can use Sapphire FL to test the interactions. So how they do is basically but you do need to probe or say to have them labeled in fluorescence, okay, in close proximity, then they will have like a probe, then they will have fluorescent signals. If they have fluorescent signals, then you can scan with Sapphire FL. So the idea right now is not whether the equipment can scan or not, because the equipment is capable of scanning anything up to 384 well plate reliably and also with a clean background, separately at 5 micron resolutions. So the thing you might need to do right now is actually to check do you have any assays allow you to probe or label your viral protein and then to probe your uh your your antiviral drug. That's the thing, because you need to visualize the interactions. So once you solve that, okay, the labeling, the probing, okay, then you can then whenever which equipment can scan, that will be a different story, because then you can option to choose which resolutions you go for. Uh, there will be very quick and easy is we just look at the signals. If there is fluorescent signals, there will be fluorescent signals. Then you definitely have a control that is without interactions, right? Then that will serve as your negative control. Then you have a positive control. Then you work based on that. Of course, yes, if you have equipment allows you to scan reliably, means scan the signals, fluorescent signals, for your negative and your positive, separate the signals very nicely. Then you can start to optimize your labeling on your antiviral drug, your labeling on your viral protein. or even your antiviral drug targets. You If you can't label the drug, you label the targets of the drug that will go to, I don't know, either be, reducing the the protein of some sort. If you cannot go direct labeling, then you go for indirect labeling of the in means you're not labeling you're labeling how to say, it's a different markers then. Okay, so right now is you need imaging system able to separate your positive and negative and your unknown signals. Then you need to optimize your labeling. Okay, right. So that I think I'm not. I think I will let you know about the application notes that will be beneficial for you. Okay. Thank you, Miss Hanis. Hey, we have another Hanis.
Hi, Kailing. Yeah, so we have another question from Dr. Hanis. So Dr. Hanis is from UPM.
Mm,
So Dr. Hanis is asking, how would you compare roughly the overall cost to detect expression levels of a specific protein using traditional Western blot versus in-cell Western? And also, Dr. Hanis would like some clarification. Can in-cell Western be performed without the Sapphire FL machine?
mm-hmm. Okay. Right. So thank you, Dr. Hanis, for the questions. So there are two here. So the first thing is in cell Western, how can you compare roughly the overall cost, the expression and expression level protein? Okay. Right. So here the I think the cost is, I would say the cost is roughly the same, because it was slightly higher because first thing is your secondary antibody. Okay, in inside Western, it is most likely that you're not able to reuse it. Okay, that is for, unlike our traditional Western blot, you can't really reuse your secondary antibody. You might be able to reuse your primary, but I would say you can do a spin down to clean it if you want to reuse it. So do your own SOP, how many times it can be reused, and then before you discard it, okay, then that will be your primary antibody and secondary antibody parts. And for the overall cost for per plate, I mean, if you've been doing your ICC, is basically that is what you have. If you have your primary antibody ready, it's basically the same. You just do not need the gel SDS page. You don't need the ECL substrates. uh Everything relies on the fluorescent signals, but you do need a system to image your fluorescent signals, so right. So the overall cost of detecting, I can't really tell you the overall cost, but I would say compared to traditional westerns, uh plus minus it will be roughly the same. So every plate versus the in-cell westerns, okay, then it will be roughly the same, because you are seeding the cell. If you have your blocker, you have your permeabilization or fixation. If you've been doing ICC, it basically is the same, a similar buffer, the similar reagents. Yeah. But if you're not having ICC, doing ICC techniques in your laboratory, then the fixing you can all self-made. They are basically, yeah, you can just punch through yourself lah, basically. Yeah. So it's like your normal westerns. For incubation with antibody, what's the volume required per well? So it's depending on the well. If you're 96 wells, so the minimum volume will be 100 micron, 100 microliter. Okay. So if a 384, there will be more or less, lesser, okay, maybe 30 or 40, that will be even good enough. Yeah. So that is for the volume. That is depends on the wells, okay, the well sizes, the incubation of primary antibody. So the idea is always try to be 5 times more than volume of the cells. So if the cells is I think 100 micron is more than enough. Yeah, 100 microliter for 96 well plate is okay. Yeah, if you worry, you can up to 200. But if you're 200, then you need to consider to reuse the primary antibody. Yeah, or secondary antibody. Yes. Reduce further. No, we don't recommend to reduce because you want the solution to be to cover the entire well properly. So you want the well because it's the idea is like this. You have those well, right? You have 96 well plate. Every single well is a round shape. So you have cells at the side, you have cells in the middles. So you try to cover the entire wells, okay, rather be a middle or the side. So if you have lesser than that, okay, I'm not sure what's the minimum you can get for 100. Yeah, so you might need to just around. If you can do your own testing, okay, if you want to reduce it, I mean, anything, all we're talking here is basically theory. All matters is go to do in your lab empirically, to perform it practically. So you have, let's say, you do a 96 plate, see your cells, okay? Then you try different reduced amount of your primary antibody, okay? But retain the secondary antibody, okay? And see whether the signals are sustained. And don't forget, you need a normalization control about the cell number. DAPI works as well, okay, or the anything that you want to count the cells, and then you see how much lower you can go, and of course, don't forget, you want to have a leeway, a buffer in case there are pipetting error or maybe something stuck in the tips or something. Just give a certain buffer volume. So with that, you're able to nail down how much lower you can go. My recommendation is we always go full out, 5 times the volume of the cells, because we do not want to miss. Yeah. But for your case, if you want to reduce the volume, do the empirical manners, detect it, do it practically, then you decide which volume you go for. Okay. That will be the best. Right. Uh I think that should be answer the question. Then I think one of the question will be, can it be performed without the Sapphire FL machine? Okay, so the Sapphire FL machine is an imaging system. So if you have any imaging system that can scan the fluorescent signals in your in-cell Western, okay, able to separate the wells nicely, okay, because you have 96 well plate, right? 96, you have separate them nicely, okay, and the signal detection is nice and a linear manner, that will be good enough, okay. Sapphire FL is something that we will recommend if you do in-cell Westerns, and if you do the 384, the only thing you can go is Sapphire FL, because it can detect, it can separate the wells to wells very nicely. Yeah, 96 wells, depending on the equipment you have in your laboratory, you can try and error. Okay, if you feel you don't like the resolutions, okay, you want the uh background is too high, you don't like it, then you might need to opt for different system. Perhaps you can try Sapphire FL. Yeah. So Sapphire FL is what we propose because it has a very clean background. Okay, it gives you 5 micron resolutions. That is we propose. Okay, that is what we propose. But you work what you have in the laboratory. Okay, then try and error. For now, is always try and error. Everything goes back to the empirical manners. Try it, done it once, test it. If you're happy with the result, you're able to analyze, plot a standard curve. That will be good enough. That is the final thing. Yeah. I hope I answered your question, Dr. Hanis. Yeah. Right. Thank you. All right. So there's a Miss Hanis here. Another question is, in terms of determining the in vitro protein ligand interaction, what's the difference between Sapphire and surface plasmon resonance? Higher detection efficiency, surface plasmon resonance. Okay. SPR. Miss Honey, unfortunately, I'm not really expert in SPR, so I can't really tell you the difference in that. This, can we let us go back and do some studies, and we contact you on this manner. Is that okay? Yeah, because I can't give you a definitive answer right now. Okay. My apologies about that.
Um, I just would like to know, Mr. Habibu, do you have any question? Because you were unmute. Do you have any question for us? Yeah, I think he mistakenly unmute himself. So if we don't have any more questions, it's already 12, nearly 12:30. I think it's quite a lengthy talk now, but I'm so grateful with all the questions from Dr. Hanis and Miss Hanis. I appreciate it so much. But just for your information, we will publish this video. We recorded it and we will publish it on our YouTube channel, but might take some time, need a little bit of editing to do. Otherwise, if you need any questions or you want to talk more on the Sapphire and the application, Fatin can go and meet you guys together with Dr. Kainan to discuss further on your applications and how we could offer the solution to you. So otherwise, I would like to close this presentation and this webinar by saying thank you. And I hope you have a great day ahead and let's talk soon. Thank you so much, everyone. Thank you, Kaining.
Thank you, Fatin. Thank you, everyone.